presentation and talk to you about the next levels of the brain, the study of the brain. So we talked about molecules and ion channels. Then Sejal talked about neurons and how they connect. So now we're going to go up to networks. So what we call networks is basically a big ensemble of neurons that connect together and they send each other action potentials, as you can see here. So in your brain, there wouldn't be a big gap between neurons. There would be glial cells to like sustain neurons and support them everywhere. So you have neurons and then you have synapses everywhere between the neurons that connect together. But then networks are built to pursue a specific function. So basically, when you have neuronal networks in the brain and in the different brain regions, you have specific neurons with specific morphologies that have specific activities. So if we take the example of the retina, you get, for example, you see a tree. The retina is this layer of this yellow layer at the back of your eye that's going to detect the image and in an inverted way, and then send information to the brain. So if we take a look at how the retina looks like, the retina is basically a neuronal, a buildup of neurons of different shapes and sizes. And if we have a little schematic, these neurons look like this. So you have a whole bunch of different neurons that have different functions. The very important ones in the retina are the ones at the back of the retina, are called the photoreceptors. So you have rods and cones. These are the cells at the back of the cellular layer that detect light. So they detect light, they detect the image, they detect colors, and then they send all of this information up to the ganglion cells, which are these cells. And then in between, you have a whole bunch of inhibitory cells that are going to refine the message. So the message I want to give you from this image is basically that you have very different neuronal types. They're all neurons, but they all look different, and they all have different functions, and they work together in order to make you able to see. And then after these neurons, so after the retina detects the image, it's going to send information to your visual cortex. If you remember, Sejal talked about the different areas of the brain. So your eye would be here, and then the visual cortex is at the back of your brain. And that's where the information is going to be computed, is going to be interpreted, so that you can finally see the picture of the tree upside down. No, actually upright. So the visual cortex is this place. And if we look, so now when the retina connects to the visual cortex, we go from one network to another network, so the retinal network to the cortical networks, and now we call that a circuit. So different areas of the brain working together in order to make you pursue a function. And so if we look at our little schematic again, the retinal ganglion cells are the one that project their axons to the back of the cortex and make a link with the visual cortex. Another view is if you cut your brain this way, or someone else's brain, I guess, this way, you can look at this is the retina, this is the optic nerve. So part of the optic nerve is going to cross to the different, so we have two hemispheres in the brain. Part of it is going to cross to the other hemisphere and then go to the visual cortex through the thalamus, which is another structure. And if you look at the motor circuit, for example, it's the same principle. So you have specialized, very long neurons that come from the brain, which would be here, go down all the way to muscles, which would be here, and their morphology serves their function. So now that we talked about neuronal circuits, we integrate each individual neurons that Sejal was talking about into a bigger picture, and these neurons communicate with each other using synapses. So if we go back to actual synapses, we can measure neuronal circuit, neuronal network firing. How do we do that? Well, basically, so if we talk about the synapse again, you know now that synaptic neuron, if neuron A wants to talk to neuron B, the neuron A is going to fire an action potential, release neurotransmitter. Neurotransmitters are going to bind to the receptors, and then we're going to have ions going into the cell. We're going to have depolarization, which means a change in the membrane potential. And this change in membrane potential is going to activate what we call voltage-gated calcium channels. These channels open when the voltage of the membrane changes. So basically, once you have the sodium ions coming in, those are going to open and let calcium in. Now, why do I bring calcium in here? Because calcium is sort of the currency of the brain. Once you open these channels, calcium flows in, And a whole lots of things are going to happen to cell B now that it received the message. And one important and very interesting thing about calcium is that it allows us to detect firing within a network. So if you have a network of neurons that are living together, for example, in a dish in vitro, you can basically see the neurons light up because the calcium is going to light up. So every time you see the light come up, it means that the neurons are firing, the neurons are active. This is the calcium that's fluorescent. So what you see, and what's very interesting, is that when you have one neuron fire, actually the whole network lights up. So one neuron talks to another neuron, but they're all connected, so everyone is going to receive a message. All right? Is that clear? I don't know if it's not. It's very complicated, I know. I don't get the part about drugs, you know. It's less interesting, I know. All right, so now calcium is also very important for plasticity. So we've been talking a lot about just neurons sending information to each other, but this is not everything that the brain does. Your brain allows you to remember things, your brain allows you to learn things, your brain allows you to process new information. So how does it do that? It does that through plasticity. So basically the networks of synapses are plastic, which means you can add and remove synapses according to what you want to do. Basically the way, an easy way to explain it is that if you want to go from let's say point A to point B in a city, what you're going to do is like build a lot of roads and a big amount of roads to go from one point to the next. But if you're never going to point C, you're going to destroy the roads and not use them, so they're going to go to waste. So it's the same in the brain. If you use synapses a lot to learn a task or to do something, then they're going to get stronger. And if you use other synapses less, then they're going to get destroyed so that they can make space for other things. So this is what's called synaptic plasticity. And calcium in cell B is going to mediate synaptic plasticity. So if cell A fires a lot to cell B, then cell B is going to change the receptor composition in order to respond better to cell A. So synaptic plasticity, as I mentioned, allows you to do a lot of things. For example, master new skills, adapt to your environment, learn a new language, or forget unimportant details. Because you also have mechanisms to forget in your brain. If you don't need to remember something, then it's going to be deleted. So this is broadly how networks work. So the cellular activity, as we saw just before with the calcium, is going to be very important to regulate networks, and then networks regulate circuits. So the way the different parts of the brain connect regulate the circuits. But now one of the big questions in neuroscience is actually to discover if we can relate the circuit to the function, which means if we can modulate circuits and then act on behavior. So can you modulate the function by modifying neuronal networks is the question that we're asking here. Because, you know, anyways, so we can't do that in human beings, sadly, but you can do that in a whole lot of other species. So we've tried, the people have studied modulating activity of the brain of other animals to see if we can trigger a different behavior by modulating the brain activity, and thus link all of this to the actual behavior that we can observe. One way to do this is optogenetics. So I don't know if you've heard about optogenetics, but optogenetics is based on the discovery of an ion channel in an algae. So now you know what ion channels are. They're basically little holes in the membrane that open under certain occasions. For this ion channel, the key to the lock is not a neurotransmitter or voltage like the calcium voltage gated channels. It's blue light. So if you shine blue light on those ion channels, then they open and they let in ions. And this way you can basically activate your cell. So what people did is take those channels, put them into neuron, and this way when you shine blue light on the neuron fires, and you can control the network. Now, this is pretty. What do we do with this? Well, you can try to put it in a model and see if you can do that at the organism level. So let's take the example of the fly. The male fly has a very specific behavior, which is grooming. So it basically cleans itself to be pretty. So it cleans the mouth, the head, the eyes. They can tilt their head pretty nicely. Look at this. Okay. So once it's so this is called the grooming behavior. People have studied grooming behavior and discovered which neuronal network controls grooming behavior. And then what people have done is basically add the channel rhodopsin, the ion channel that if you shine blue light on it, it opens. They added that to the neurons that control the grooming behavior. So what do you think is going to happen when you shine a light?
Groom.
Is it really? So the light is going to come from the bottom right here. Yeah. So basically, every time you shine a light, the Drosophila is going to start grooming, and when you stop, then it stops grooming. So you're basically controlling behavior through controlling networks. And it all started with ion channels, so at the molecular level. Do you see where I'm going here? This is crazy, right? All right, so now big question, why are we doing this? Why are we trying to make flies groom more? So this is not everything, we do more than that, we do a lot of things. But the reasons why we're doing this is because understanding neuronal networks in those very basic models, such as Drosophila, and then in the mouse, and then in the monkey, allows us to later relate the information to human brains and see how the human brain works. And once you understand which parts are connected to which parts, you can fix a brain that would be broken. For example, if you take this lady is a tetraplegic, so she's not able to move her limbs, but her brain functions perfectly. So the only problem here is that her brain cannot send messages down to the rest of her body. But since we know now, because we studied neuronal networks, that this part of the brain, which is here at the top of your head, controls hand and arm movement. Basically, researchers have recorded the activity in her brain, translated this activity into a machine that will do the movement for her. So she's thinking about grabbing the coffee right now. And this is actually the first time that woman can drink a coffee by herself. So there is a point to all of this, and I think this is very interesting and very brilliant technology that could be used in the future years. So yeah, basically that was my part. The take-home message, so if you need to remember one thing from everything we said today, neurons are the basic units of the brain. They connect and they form networks and circuits, so that we study networks and circuits in order to understand how behavior works, how the organisms work, and then manipulating the circuits can modify the behavior, like we did in the fly, and understanding those circuits can help develop new treatments and cures for people that have defects in translating the information from the brain to the rest of the body. I have a little bonus for you. So if you have one thing that I want you to remember and also spread, it's that, did you hear about the 10%? We only use 10% of our brain? This is not true. No. So please tell everyone that we use 100% of it, because some people doubt it. Don't watch movies. So we've seen the different parts. Every part of the brain has a specific function. Every cell is useful. Everything's useful. If it wasn't, then you would get rid of it. So yeah, if you can spread that word, it would be great. And finally, I want to thank our partners, so Concordia, McGill, the Brain Program, CAN, the NUHC, and of course Christian for organizing all this. And that's it.